So hello everyone, welcome back to Hasvora Education. Today we will be solving the Biology 9700 Paper 2 Variant 2 of the May-June 2025, and the time allowed for this paper is 1 hour and 15 minutes, and the total marks for this paper are 60 marks. So this is the latest paper that we have for the theory of the Biology Paper 2. So let's get started with the first question, okay? So the first question says, root hair cells are specialized plant cells located in the outer layer of the young roots of plants. Root hair cells have an essential role in the uptake of water and dissolved minerals, ions and salts from the soil solution. The transport of water across the root to reach the central xylem tissue can occur by the symplast or the apoplast pathway. So you should know the difference between the symplast and the apoplast pathway. The symplast pathway actually involves your cell surface membranes, and the apoplast pathway it does not involve your cell surface membrane. It involves your intercellular spaces and your cell walls. Okay, so Figure 1.1 is a diagram of photomicrograph of a root hair cell. So you just have to label these cell structures. So first of all, the X that we have, this is pointing towards this area, which is actually your vacuole, but some of the students might confuse it with the contents of the vacuole and might write it as sap vacuole, but here they are asking you the cell structures. Okay, so this is your vacuole. Then for Y, this is the layer that is actually encapsulating the vacuole, and this is what we call the tonoplast that layers the vacuole contents, like the sap vacuole, which contains the amino acids, the carbohydrates. So it actually layers it. Then for the Z, we can clearly see this is the nucleus. Okay, so this is your nucleus. Let's move on to the second part. So in the symplast pathway, water passes through the cells of the different tissues in the root before entering the xylem vessels of the xylem tissue. So we know that first of all, when the water is absorbed from the root hair cells, it moves to the epidermis, then into the cortex cells, and then into the epidermal into the endodermal cells, and then through the cibrin, where we have actually the hydrophobic layer, after which some of the materials are selected, and then it passes to the xylem. Okay, but here they are asking that name the tissues, so we have to name the tissues. We do not have to give an answer in the form of cells, okay, because you will lose your marks if you write cells. So in the correct sequence through the path which water passes in the symplast pathway. So first of all, you will write that water passes from the epidermal tissue, which is the outermost tissue, the epidermal tissue, to the cortical tissue. You could simply write the cortex cells, but here we have to mention the tissues, and then it moves to the cortical tissue, to the pericycle via endodermal tissue, which actually contains the endodermis. So the endodermal tissue to move into the xylem. Okay, so we have actually just correctly sequenced all of them. So first of all, it moves to the outermost tissue, which is the epidermal tissue, and then into the cortex cells, the cortex tissue, and then into the pericycle via the endodermal tissue, in which some of the materials are selected because the apoplast pathway is just hindered at that way. Okay, so the things that can only move to the symplast pathway. So this was the answer for this question. Let's move on to part C. So in the apoplast pathway, water passes along the cell walls of the adjacent plant cells and through the intercellular spaces. This is more efficient than the symplast pathway. So explain the structural features of plant cell walls. So we have to list the features of the plant cell walls that make the apoplast pathway an efficient pathway for the transport of water. So actually we have to state that how it accommodates, or you can say how it supplements the water in moving through the to the apoplast pathway, how the features of the plant cell walls supplements it. Okay, so first of all we can write about the OH groups in the cellulose molecules. That OH group, they are actually called the hydroxyl group. They are hydrophilic in nature, and anything that is hydrophilic, that makes the hydrogen bonds with the water molecules, and that can actually you can say form a type of water channel and provide a smooth pathway. You can say it makes a smooth channel for the water flow. And another thing is that there is spaces between the cellulose molecules, so that actually provides no hindrance to the water flow. Okay, and the third point that we can write about is that the cellulose molecules are hydrophilic. The first one is the cellulose molecules are hydrophilic. The second one is about that there is no hindrance, and then the third one is that it is fully permeable to the water. Okay, so it provides no hindrance to it. So you can write about this, right? So the cellulose molecules has hydrophilic OH, or you can say hydroxyl groups, which make hydrogen bonds with water and form adhesive forces. Okay, so we actually call it the adhesive forces. So forming a water channel, or you can say a water flow. Okay, and after that we can write about that there is no hindrance. So the separation between cellulose layers, because they are arranged in the parallel sequence, so the cellulose layers have some space, and that's why the water flows between them. Okay, so the space between the cellulose layers allow the water to flow. And third point can be we can write it as the cellulose, or you can say the cell wall, actually the cellulose cell wall, or you can write it as cell walls. So the cell wall is permeable, fully permeable, so it provides no hindrance or obstruction to apoplast pathway. Okay, so this will be your answer for 3 marks. We have listed 3 points. First is for the hydrophilic OH groups. So we have stated that it makes hydrogen bonds, it eases the formation of water flow. So, and the second is the separation, which allows the water to flow. And the third one is that it is fully permeable, so there is no obstruction or you can say hindrance in the path of that apoplast pathway. So let's move on to the D part. So root hair cells are also synthesized and secrete substances into the soil. Electron microscopy of the structure of the root hair cells are identified endoplasmic reticulum. So a number of small Golgi bodies and numerous vesicles are present in the root hair cells. Okay, so root hair cells of the barley plant secrete enzyme known as acid phosphatases. So this is your enzyme, and which catalyze the release of inorganic phosphate ions to form the organic phosphates in the soil. root hair cells of the sorghum plant secrete a hydrophobic lipid compound known as the sorgelin, which slows down the growth of neighboring plants. So, explain why the proportion of rough endoplasmic reticulum or to smooth endoplasmic reticulum may differ in the root hair cells of the barley plants compared with the root hair cells of sorghum plants. So, first of all, let me explain it. So, we have read that in the barley plants we are actually synthesizing what we need, the enzyme which is called the acid phosphatases. So for the enzymes we know they are globular proteins, and for them we need to have the endoplasmic reticulum. Okay, and while for the lipids, the lipid synthesis occurs in the smooth endoplasmic reticulum. So that is why the smooth endoplasmic reticulum will be in high proportion in the sorghum plant, because we need the lipid compounds, okay, which is, you can say, the sorgelin. And in the you can say the barley plants, we have the enzyme, we need the enzymes, so we are actually needing more proportion of the rough endoplasmic reticulum for the synthesis of your proteins. So this is what you have to write. 1 point for the barley plants, that why they have, and 1 for the sorgelin. So you can simply write it as the barley plants have high proportion of rough endoplasmic reticulum, as they need more site of synthesis of proteins. And what are the proteins that we need are the enzymes. Okay, you can just write this in the bracket, so they know that you are referring to the enzymes. Okay, so we need more site of synthesis of the proteins, while sorghum plant needs more synthesis of lipid compound, hence it will have more proportion of smooth endoplasmic reticulum. Okay, so this will be your answer. Let's move on to the part 2. So student X stated that the acid phosphatases and the sorgelin could be transported out of the root hair cells using the same process, and the student Y stated that the acid phosphatases and the sorgelin are transported out of the root hair cell using a different process. Okay, so these are actually two students. They are giving the hypothesis that one is saying that both of the molecules are transported using the same system, and the other one says that it is being transported out of the root hair cells using a different process. Okay, so suggest reasons given by the student X and the student Y to support their statements. So we have to support both of the cases. So how can we just think of it like that you can say that acid phosphatases, it is an enzyme. Okay, so this is actually your enzyme, and enzymes are actually globular proteins, and the proteins will have, you can say, the charged amino acids, and they are actually not hydrophobic. Okay, the enzymes are hydrophilic. So we have studied that whenever we have hydrophilic molecules, or you can say the ions that are charged, or you can say they are large in size. Okay, so they needs to be transported using the bulk transport, or either you can say using the active transport. Okay, so first of all we can say that both of the molecules, for supporting the verdict of the student X, we can say that both of the molecules are large and they are being transported in a large quantities, so that is why they will be transported using the bulk transport. Okay, and for the second one, we can say that the sorgolene, as they have also told us in the question, that they have told us that it is a lipid, and we know that lipid is a hydrophobic. By the way, they have also told you about this, that it is a hydrophobic lipid compound, and lipids can be directly transported using the diffusion, okay, down the concentration gradient. So for the student Y, we can say that the acid phosphatase is a hydrophilic, and it can be transported using the mechanism of your active transport, while the solgidine can be transported using the diffusion, as it is a hydrophobic substance. Okay, so we'll write the answer for this. So for student X, we will say both of the molecules which are the acid phosphatases and sorgolene are large in size and transported in large quantities, so they use bulk transport, okay, and exocytosis. Okay, so they use exocytosis because they are large in size and they cannot be transported directly. While for the second one, we can say that since acid phosphatases is a hydrophilic substance, or you can also write it as hydrophilic enzyme. Okay, so you can write it is a hydrophilic enzyme which can't move through the phospholipid bilayer directly. So it is transported using active transport. And for the second one, while sorgolene is a hydrophobic compound, so it moves directly via the phospholipid bilayer down the concentration gradient. Okay, so this is how you will actually support both of the verdicts using the transport mechanism and using the knowledge about the molecules that you have. So this was for 4 marks, and let's move on to question number 2. So the transport of respiratory gases involve the blood plasma and the red blood cells. Red blood cells contain the globular protein, which is the hemoglobin. Describe the features of a hemoglobin molecule that are typical of a globular protein other than having an approximately spherical shape. Okay, So we have to give the features that are actually present in most of the globular proteins and that are also present in your hemoglobin protein. So first of all, let's talk about the spherical shape. Okay, so we do not have to state it directly, but we have to use the structural feature. Like for example, for the spherical shape you need to have a tertiary structure. Okay, so first of all, for the globular protein you should have a tertiary structure, because it cannot be it cannot form a globular protein until you have a quaternary structure or your tertiary structure. And the second one is that the globular proteins are soluble, so that's why it has the hydrophilic R groups that are pointing outwards and the hydrophobic R groups that are pointing inwards. So these are also two of the features, so you can write any of them, any two of them. So first of all that I will write is they have hydrophilic R groups pointing out of the core. Okay, or you can say out to the aqueous environment. So let's write it like that. So it will be pointing out to the aqueous environment. While you can also write it as they have hydrophobic R groups pointing to the core. Okay, so both of them, the first and the second statement, are actually the cross arguments of each other. And the third one will be that they should have a tertiary structure. So these are actually your 3 points for the globular proteins. The first one is for the solubility, because they are soluble, so they have the hydrophilic R groups pointing outwards. And the second one is the cross argument of that. And then the third one is that they should have a tertiary structure, because they have a spherical 3D shape. That is why they should have a tertiary structure or your quaternary structure. Okay, so this will be your answer. Let's move on to part B. A number of substances are involved in the transport of respiratory gases. Complete table 2.1 by stating the names of the substances that matches the description of its role in the transport of respiratory gases. So this table is actually related to all of the transport mechanism of the respiratory gases. Like for example, the carbon dioxide, it dissolves in the water to form the carbonic acid, and then it dissociates to form the carbonate and the hydrogen ion and all of that stuff. Okay, the chloride shift, and then the formation of the carboamino hemoglobin and the oxyhemoglobin, and then how the affinity for the hydrogen ion is more, and then how the oxygen dissociates at low partial pressure and the high partial pressure of the carbon dioxide. Okay, so first of all, they are filled for you is that in the capillaries of the respiring tissue, this combines with water to form the carbonic acid. So this was the easy one. So this is the carbon dioxide, because your carbon dioxide combines with your water to form this H2CO3, which is called your carbonic acid. Then for second one, it says in the capillaries of the respiring tissues, this enters the red blood cell through a membrane transport protein. Now first of all, just focus on this word. Okay, this is the biggest hint in this whole line. Okay, so it actually moves through the membrane transport protein. Now by this we know that it is a charged molecule or an ion, because only a charged molecule and ion can move through a carrier protein. And then we also have that it enters the red blood cell. Okay, so if you remember that this carbonic acid dissociates to form the H plus ion and HCO-3, which is the bicarbonate ion, and this bicarbonate ion actually moves out of the red blood cell. So to actually balance the negative charge, what actually happens that the chloride ion moves in. Okay, so this will be your chloride ion. So we can just simply write chloride ion. Then third one says in the red blood cells, this is formed when the carbon dioxide binds to the hemoglobin. So you should know this, that the carbon dioxide binds to the hemoglobin to form the carboaminohemoglobin. Okay, so most of the students confuse it with the carboxyhemoglobin. There is nothing known as carboxyhemoglobin. So when the carbon dioxide combines with the hemoglobin, it forms carboaminohemoglobin. Okay, then for the third one, in the alveolar capillaries, this combines with a hydrogen ion in the red blood cell to form the carbonic acid. Now I have already showed you the dissociation of the carbonic acid. So this forms the hydrogen ion plus the bicarbonate ion. Now what they are asking you is that what combines with the hydrogen ion to form the carbonic acid. So it is your bicarbonate ion. Okay, you can decide it as the carbonate ion or the bicarbonate ion. So this is your answer. Let's move on. So in the alveolar capillaries, this is formed in the red blood cells as the result of oxygen binding to the heme group. Okay, so whenever oxygen binds to the heme group, it forms your oxyhemoglobin. Then for the last one, in the red blood cells, this combines with hemoglobin to form hemoglobinic acid. Now you should know that the H plus ion has more affinity for the hemoglobin than the oxygen. So whenever there is a low partial pressure of the there is low partial pressure of your oxygen and high partial pressure of the carbon dioxide. So the carbon dioxide first of all forms the carbonic acid, then dissociates. Then this hydrogen ion that is formed after the dissociation, this has higher affinity for the hemoglobin molecule. So it dissociates. It actually, you can say, releases its oxygen and it binds with this H plus ion. So the H plus ion combines with the hemoglobin. So this forms your hemoglobinic acid. Hemoglobinic acid. Okay, so this will be your hydrogen ion. Then let's come to the third question. Okay, so carrots and roots are roots vegetables of the carrot plant. The carrot plant is an important food crop that is grown throughout the world. Carrots have a sweet taste because sugars form a proportion of the total carbohydrate present. Plant breeding has produced many different types of carrot with different level of sweetness. So one of the sugars found in the carrots is galactose. Galactose has the same molecular formula, and by the way, you should know that all of the monosaccharides have the same molecular formula as the alpha glucose. So but their arrangement is a little bit different. Okay, so figure 3.1 shows the molecular structure of the galactose that is found in the carrot. This is similar but not identical to the molecular structure of alpha glucose. So you can see that here we have actually the OH groups are inverted. So state the differences between the molecular structure of galactose shown in the figure 3.1 and the molecular structure of alpha glucose. Now this is worth of 1 mark, and this is actually a very simple question. So first of all, if you know the structure of the alpha glucose, let me just draw it. So this is your structure for your alpha glucose. So it is something like this. Okay, so this is your structure, and we have the oxygen here, then we have the CH2OH, then we have the OH pointing downwards because this is the alpha glucose. Okay, and remember in the alpha glucose we have the OH pointing downwards, so it is downwards light here as well, and we have H here, then we have OH and H, and then again we have OH and H. Okay, so this is a structure like this. So first of all that we can notice is that the OH is actually inverted. Okay, this is inverted, this point. So you can just simply write it like this, that the carbon number, the groups that are attached to the carbon number 1 and the carbon number 4 are the other way around, or you can say that the hydroxyl groups that are attached to the carbon 1 and carbon 4 are pointing downwards in your alpha glucose molecule. So this is what I'm going to be writing, is that the hydroxyl, the OH groups attached to carbon 1 and your carbon 4 are pointing downwards in alpha glucose. Okay, so this is your 1 mark answer for this question. Let's move on to part B. An investigation was carried out to determine the sugar content of the mature carrot plants produced by different local varieties of carrot plants that are grown in Tunisia, the north of Africa. So carrot plants were grown from seed under standardized conditions. When mature, the carrot were harvested. The different sugars present in the carrot were identified. Measurement of the sugar content of each of the sugars were made, and table 3.1 shows the result of for 5 different local varieties of plant, the carrot plant, which is from A to E. So after studying the results shown in the table 3.1, a student concluded that the carrots from the local varieties contain the same 4 sugars, and the student made 3 conclusions. Okay, so he made 3 conclusions, and the conclusions are that the first one is that there are non-reducing and reducing sugars in the carrots. The second one is that there are monosaccharides and disaccharide sugars in the carrots, and third one is that the carrots have a same pattern of results. Okay, so these were the 3 conclusions. Let's just see what the examiner is actually asking us about. So explain that evidence in the table 3.1 supports these 3 conclusions. Okay, so first of all, the first conclusion that he has given is that there are non-reducing sugar and the reducing sugar that are present in the carrots. So first of all, what are the non-reducing sugars? You only have one non-reducing sugar, which is the sucrose. Okay, and the reducing sugars are the galactose, your glucose, and you can say the fructose, and the other 2 disaccharides, which are actually your lactose and the other one as well. Okay, so these are actually all of your reducing sugars, and only there is one non-reducing sugars in your syllabus, and that is your sucrose. Okay, so you can see that all of these carrots from A to E, all of these contains your fructose, galactose, glucose. By the way, these 3 are your reducing. Okay, these are your reducing sugars, and only sucrose, which is the non-reducing sugar, is also present in all of them. Okay, so you can just support this first conclusion by writing that fructose, you can just write it as this. Okay, so reducing sugars like the fructose, glucose, and galactose, and then the non-reducing sugars are present in all varieties of carrots. Okay, so we can just see it, and we have actually named them and specified them into the groups, that these are the reducing sugars and these are the non-reducing sugars that are present in all varieties. Okay, so the second conclusion, let's see what the second conclusion was. So there are monosaccharides and disaccharide sugar in the carrots. Now out of the 4 of the sugars that they have given us, the fructose, the galactose, and the glucose, these are actually your monosaccharides. Okay, and the sucrose is only your disaccharide, and the sucrose is actually your only disaccharide. So we can just simply write it as monosaccharides such as the fructose, glucose, and galactose and disaccharide, which is only the sucrose, are both present in all the carrots. Okay, then let's come to the third conclusion, which is the carrots have the same pattern of results. Now, what do they mean by the pattern? So, if you see that for the local variety of the carrot A, so if we see that the galactose has lower content than the sucrose, then the sucrose has the lower content than the glucose, and then the glucose has lower content than the fructose. Now, if we see for the second one, for the B, it is the same pattern. Okay, so the sucrose is less than your glucose, and then the glucose is less than your fructose, and the minimum is your galactose. Okay, so first of all, what you will write in your answer is that the pattern of the sugar content in all of the carrots is same. The sugar content of the galactose is the minimum, followed by the sucrose, you can say the glucose, and then your fructose. Okay, so what you can write is as the pattern of sugar content is same, highest sugars the highest proportion of the sugar is your, let's see what it was. So it was actually your fructose. So highest proportion of the sugar is fructose, and the lowest is your glucose, I think. Okay, so it is glucose, so the lowest is glucose. Okay, so the pattern is same for all of them. Then the C part says carbohydrate that are not sugars are also present in the carrots. Name one carbohydrate that is present in the carrots and not the sugar. So you can write about the starch. You can also write about one of the component of the starch, like for example your amylose. You can also write about the amylopectin. You can also write about the cellulose because it forms the cell wall. But these are actually not the sugars. Okay, they are not sugars. So you can write it from any of them. So then let's come on to the D part. So each carrot plant produces a carrot in the first year of growth. If the carrot is not removed from the plant after it matures, the plant passes through a dormant period and the shoot develop from the carrot in the second year of growth. So first of all, what they have said that when a carrot is produced, if it is not removed within the period, then after the dormant period starts, the shoot develops from the carrot. Okay, so what it means, when the shoot develops from the carrot, this means that it starts using the nutrients from the carrot, and it actually uses it as a source. So this allows the flowers to be produced and the seed formation to occur before the plant dies. So explain with reference to the life cycle of the carrot plant, when the carrot acts as a source and when it acts as a sink. So first of all, what we can write it that when it actually behaves as a source. So it behaves as a source when the shoot develops from the carrot after the dormant period. Okay, because it starts to, you can say, it takes the nutrients from it, and it actually the assimilates, tries you can say, are transported to the growing regions. So you can write it as the carrot plant acts as source after the dormant period as the assimilates are converted, like what is stored in the carrot, it is converted to your sugars from the starch. It is converted to the sugars and transported to the growing regions. Okay, so transported to the growing regions. For example, the assimilates also contain your amino acids, so they are needed at the growing regions. So because the cell content is made from the protoplasm, okay, and the main component of it is your amino acids, so they are transported to the growing regions. Then let's come on to the sink. So it behaves as a sink before the dormant period when it is growing. So before the dormant period, carrot acts as sink for storage of assimilates okay, so it is used as a storage site before the dormant period when actually it is you can say behaving as a root. Okay, so that is when it is actually behaving as a sink. So all of the assimilates, or you can say it moves from the site of the photosynthesis. Whenever you can say the carbohydrates, the glucose are actually manufactured, the sugars are converted to the starch, and then it is being stored in your root, which is the carrot here. Okay, so the assimilates move from the site of synthesis. So site of synthesis is your leaf in most of the cases in your photosynthesis. Okay, so it moves from the site of synthesis and are converted to starch for storage in carrot. Okay, so it acts as a storage space. Let's move on to the part E. So carrot virus Y is a pathogen of carrot plants. The virus, which belongs to the group known as the potyvirus, and replicates its viral nucleic acid proteins within the host carrot cells. The general structure of the potyvirus is shown in the figure 3.2. So you can see this is the structure. It actually has the RNA, and this is the protein coat, or which you call your capsid. So the synthesis of the viral proteins in the host carrot cells only involves the process of translation. Now translation is actually forming of the polypeptide chain of the protein, and the process of the transcription does not occur. So suggest why translation occurs in the host carrot cells during the synthesis of viral proteins, but transcription does not occur. Now what is transcription? Transcription is whenever for example, if you have a DNA, then you are transcribing one of its strand. You are actually copying the same code into one strand, okay, and then that one strand is actually translated and it forms a polypeptide chain. So in this case, the reason why the transcription does not occur is that because you only have this RNA, while your carrot has your DNA. Okay, these carrot plants actually has your DNA, so you cannot transcribe from a DNA, and this RNA is actually used as a mRNA. So mRNA does not need transcription; it is just directly translated via the. It is just directly transcribed. It is just directly translated via the ribosomes that are already present in your host cell. Okay, so you can just simply write it as the viral RNA acts as mRNA and is directly translated via the ribosomes present in host cell, because the ribosomes are always present in the cell. Okay, so via the ribosome present in host cell, but it cannot be transcribed from a DNA. Okay, so how can you transcribe RNA from a DNA? Okay, so basically they do not match. Okay, so that will be your answer. Let's move on to question number 4. So question number 4 says tuberculosis is an infectious disease that is caused by a bacterial pathogen. The pathogen has a mechanism to avoid digestion by phagocytes, macrophages, and may engulf the bacteria, but in some cases the bacteria remain alive within the cell instead of being killed. So although there are 2 main species of the bacterium causing TB, it is rare for the species that causes the bovine TB. So actually the pathogen for that one is actually called your the Mycobacterium bovis. So this is actually your bovis variant. So this is called the bovine TB in the cattle to infect humans. So actually they are saying that it is rare for the species that actually cause the TB in the cattle to infect the humans. Okay, so name the species of the bacterium that is the main cause of TB in your humans. So the bovis variant actually causes the TB in your cattle. So in the humans we have Mycobacterium tuberculosis. Okay, so this is the variant that causes the TB in your humans. Then B part says explain how the pathogen name in A is transmitted from a person with the disease to a person who is uninfected. Okay, so actually we have to tell the mode of transmission. The first mode of transmission is using the contaminated areas. For example, if there are airborne droplets, or you can say the mode of aerosol transmission. For example, if a person is actually sneezes or coughs, then the aerosol droplets will actually have that bacteria and that can be transferred to another person. Then the second mode of transmission is when you eat something contaminated, like from the meat from the cattle or your unpasteurized milk. Okay, so you can write any of them. So the first one is when a person with disease coughs, the droplets in the air may contain the pathogen and can be transmitted. Okay, and the second one is eating unpasteurized meat from cattle and drinking unpasteurized milk. Okay, so these are your 2 reasons. Now the C part says state a type of therapeutic drug you should treat your TB. So TB is actually caused by a bacteria. It is a bacteria and the pathogen. So the drug that we use to treat a bacteria is your antibiotic. Okay, you can just simply write it as antibiotic, but in the case if you are confused that you have to write a specifically named antibiotic, you can use the name of rifampicin. Okay, so rifampicin is one of the antibiotics. Let's move on. So D part says figure 4.1 is the photomicrograph of a section of a lung tissue taken from a person who has not been infected with the bacterial pathogen and who does not have TB. Okay, so what we have to do is, so blood vessel and some structure of the gas exchange system are visible in figure 4.1. So on figure 4.1, use a label line and label a bronchus, a bronchiole, and a blood vessel. So first of all, let's just zoom out a little bit. So you can see these structures actually. So this is actually your vein, because I can clearly see it has a large lumen, and then this one has a smaller lumen, so this is possibly an artery. Okay, so you can just label any one of them and you can label them as a blood vessel, okay, because they are inquiring you to label a blood vessel. So this one is actually your blood vessel, and then for the bronchus, this one is your bronchus, because we know that the bronchus is actually, it is convoluted, the innermost layer is convoluted, or you can say it has folds. Okay, so this will be your bronchus, this will be your bronchus, and the one that is less convoluted, or you can say it is morely straight, so that one will be your bronchi. It will be your bronchiole. Okay, so this is how we have to label. Okay, so let's move on to part number E. In some people with TB, areas known as granulomas may form in the lung tissue as a part of immune response. Okay, so keep in mind that this is actually your immune response to this pathogen. So what actually the immune response does, that it forms granulomas. Let's see what granulomas are. So multinucleated giant cells formed by the fusion of many macrophages. So I think this is actually what it does, is that it will actually try to isolate the pathogen. Okay, like once you have studied that the agglutination. Okay, so what actually happens, that the pathogens are actually, you can say, they are isolated, or you can say they are packed together so that they can be destroyed via phagocytosis. So dead cells and infected cells, mainly macrophages, so that is what actually forms your granuloma. So the question asked, with reference to figure 4.1 and figure 4.2, describe and explain how the changes that occur in the result in the granuloma formation can affect the gas exchange. So we have to state that how it affects the gas exchange and harm the health of an infected person, and it may help to prevent TB developing in other parts of body. So we have to address both of these points. Okay, and we have 5 marks, so I will try to write 3 points for both of them. So we have to compare both of the images. So you can see that we have stained area here, so this means that the granulomas will form and it will actually damage. The one aspect is that it will damage the alveoli. So you can see that here we have the clear alveoli. So what it will does, that it will block the alveoli, and as a result what will happen, that when there is less alveoli, there will be less surface area for the gas exchange. As a result of less surface area of gas exchange, this means that there will be less oxygen taken up to the tissues and the person will feel more fatigue. So you can just simply write all of this. So granulomas. Now I'm writing about this one. Okay, I'm writing about the first point. Okay, so granulomas will damage and block the lumen of the alveoli. So what will happen when they damage them? There will be less alveoli, resulting in less surface area for absorption. Okay, less surface area for the absorption, or you can just write it as less surface area for your gas exchange. So less surface area for your gas exchange. So there will be less oxygen reaching the tissues, and person might feel fatigue, or you can say breathlessness. Okay, so breathlessness. Now let's talk about the second pointer, that how it will help. So first of all, it will isolate all of the, you can say, the pathogens, and when it isolates, it packs it, then they can be you can say recognized by your you can say their antibodies. They will be antigen presentation, and then it will be destroyed finally via phagocytosis. So we have to add the second pointer, so it will be the granulomas will isolate and prevent the movement of pathogen to other parts of body. They are packed, and you can say agglutinated, to be destroyed by the phagocytes. Okay, so they will be, first of all, they will be isolated, they will be prevented to move further, and then they will be destroyed by the phagocytosis by the phagocytes. Okay, so this is worth of 5 marks. 2 for the how we have to prevent, it will prevent a TB, and 3 for the how it will affect your gas exchange system. So the first point is that it will damage. The second point is that they will be less alveoli, they will be less surface area for the gas exchange. The third point is that how it will affect and how the person will feel when they will be less surface area for oxygen. Okay, then the fourth point is that it will isolate and prevent the movement of the pathogen, and the fifth point is that it will get destroyed by the phagocytes, that it will be recognized more by the antibodies. Okay, or you can say the white blood cells. So we have stated all of the points, and this is how the question was going to be done. Let's move on to the next question. Now question number 5 says there is a global shortage of blood for transfusions. Researchers can culture bone marrow stem cells in the laboratory to manufacture red blood cells for the potential use as an artificial blood product. Okay, so they are actually saying that they can manufacture the red blood cells using the bone marrow stem cells, and they can actually use for the people that actually have lower concentration of the red blood cells. Okay, so the researchers collect bone marrow stem cells that are present in the small quantities in blood rather than extracting them from bone marrow. Actually, so what they are doing is that they are actually using the bone marrow stem cells that are already present in the you can say in the blood, so rather than extracting it from the bone marrow. So it is actually a less invasive procedure. So antibodies specific to bone marrow stem cells are attached to the tiny magnetic beads. The beads are added to the sample of blood. An electric field is applied to immobilize the beads so that the bone marrow stem cells are then collected. Okay, so this is the procedure of collecting the bone marrow stem cells. So the antibodies are attached to the beads so that they are the sites used for binding to specific molecules on the bone marrow stem cells are left exposed. Name the term given to the specific molecules on the bone marrow stem cells that attach to the antibody binding sites. So we know that what attaches to the binding sites of the antibody, they are called your antigens. Okay, so they are antigens. Now, second part says suggest and explain the advantages of using bone marrow stem cells from the blood sample to manufacture artificial red blood cells. Now, how can we actually benefit from it? So, first of all, you can say that it is a less invasive procedure. So, because we are not directly extracting the bone marrow, but rather we are using the bone marrow stem cells that are already present in your blood. The second point that can be valid is that you can actually they are mitotically dividing, so you can use it again and again to you can say form a large number of red blood cells, and you can produce large number of genetically identical red blood cells. Okay, so 3 points will be valid. Let's just write this. So the bone marrow stem cells can be used to produce large number of genetically identical red blood cells and can be divided multiple times. Okay, then you can also write about that actually using you are actually using the bone marrow stem cells, so there is less chance of passing disease to a person. And the last point that I just told was that it is less complicated, or you can also write it as invasive procedure than directly extracting from bone marrow. Okay, so this is over for 3 marks, and this will be answer. Then C part says one desirable feature of the artificial blood products such as the artificial red blood cells is that they should be economical to produce. So first of all, suggest other desirable features of the artificial blood products. So these are actually the type of question that typically comes out, and you should remember about 5 to 7 points of this. So the other desirable features is the low toxicity, that it should have low toxicity, it should not stimulate or trigger, you can say allergy, or you can say it could not cause an infection, or you can say it could not stimulate a disease. Okay, so for example, if I'm having you can say a vaccine for a particular bacteria, so it should not have toxicity, or it should not trigger that specific disease in my body. Okay, and then the second one should be that it should have large amount of supply so that it can meet the demands of the people. Then the other one is that it should have a long shelf life, it should be resistant to temperature changes. Okay, so that it can be transported for, you can say, for a long distance. And the other one that we can talk about is that it should have all of the it should have the same effect on all of the ethnicities and the cultures. Okay, so these are the 4 points. So let's just write these 4 points. The first one should be that the product should have low toxicity. It should not trigger an allergic response. Okay, like some people are allergic to some of the vaccines. So it should cater that. And then the third one is that it should have long shelf life. It should be thermostable so that it can be transported for a long distance. And then it should work on all ethnicities and age groups. Okay, so it should have the same effect on all of the people. Then let's come on to the last question, which is the question number 6. So horseradish peroxidase is an enzyme that can be extracted from the roots of the horseradish plant. So the scientific name is Armorrhiza rustinica, and HRP is used extensively in the industry, and the technology in the reaction catalyzed by HRP, hydrogen peroxide, is used to oxidize an organic substrate. Okay, so this is the organic substrate, and this is being catalyzed by the HRP enzyme to form the oxidized substrate and your water. So inhibitors can have effect on the Vmax, okay, and the maximum rate of reaction, and the Km value, which is the Michaelis Menten constant of the HRP. So figure 6.2 shows the effect of substrate concentration on the rate of reaction of HRP. So on the x-axis they have given you the substrate concentration, okay, and this is the graph. So now they are asking us that complete figure 6.2 by drawing the curve to show the presence of a non-competitive inhibitor. Now you should remember that what a non-competitive inhibitor does, it decreases the value of the Vmax. So the Vmax value decreases, so you can just simply half the value of Vmax, and the Km value, now this is important, the Km value remains same. Okay, so you should be careful for it. Use the curve you have drawn to obtain an estimate of the Km value. Now I just told you that the Km remained the same. Okay, so let's see the Km value for this one. So Vmax, this is the Vmax, and the Km value is the substrate concentration for half the Vmax. So half Vmax will be your 4, and this is the point. Okay, this is the point, this is approximately 0.06. Now this should be the same for what we are going to be drawing. Okay, so for example, if I'm going to be drawing the Vmax here, this is my new Vmax. Okay, so I should have my Km value about here. So half of 4 is 2, so my graph should pass through this point. Okay, so this is how I will make it, and now the Km value is 0.06 millimole per dm cube. Okay, now scientists can design synthetic DNA nucleotide sequences to produce a synthetic HRP gene. These sequences will include a start codon, a stop codon, so that the translation of the messenger RNA can occur. Explain what is meant by the start codon and stop codon. So the start codon is actually the first triplet nucleotide which actually starts the translation, or you can say it is translated to the methionine amino acid, and it starts the elongation of the chain. Okay, so let's write about the start codon. So start codon is triplet of nucleotide bases that is translated to methionine. Okay, so it is translated to methionine, and it starts the elongation of chain, while the stop codon ends the chain translation, the polypeptide chain translation, and it detaches from the ribosome. Okay, so it detaches from the ribosomes. So this will be your answer. Let's move on to the part C. HRP is used in an immunological test known as a sandwich ELISA. One use of the test is to diagnose a disease. So figure 6.3 outlines the main steps in the sandwich ELISA. So it is actually an immunological test, so in which a toxin is released by a pathogen, and it is detected in a sample of body fluid taken from a person who is ill. Okay, so the test involved two types of monoclonal antibody that can bind to the toxin, a capture antibody and a detection antibody. So now let me tell you something about the capture and the detection antibody. The capture antibody, they are immobilized. Okay, so these are immobilized and fixed antibodies that actually capture that pathogen, while the detection antibody then binds to that pathogen and it actually contains the enzyme. Okay, so they contain your enzyme, but both have the similarity. Now both have the similarity to bind to the pathogen. So first of all, the step one is prepare a test well by immobilizing the capture monoclonal antibody to support the surface. So you can see that they are fixed. Okay, they are fixed to the surface. Now the second step is that remove a sample of the body fluid from a person who is ill and add the sample to the test well. So you can see that it has captured this toxin. Okay, so as I told you, this is actually your toxin and it has been captured by your capture antibody. So wash out the test well to remove any substances not bound to the capture antibody. Add detection antibody to the test. Now what this detection antibody will do, it will just bind to this. Okay, and it actually has the enzyme. So wash out a test well to remove any detection antibody that is not bound to a toxin, and add substrate for the catalysis by HRP. So HRP is your enzyme, and add acid after a set time to stop the reaction. So complete figure 6.4 to show the situation in the test well after step 5. So step 5 was actually when you have added this HRP and a detection antibody. So detection antibody has this enzyme. Okay, so it will bind to a toxin. So we just have to show that it has binded to a toxin. You can just show two of them like this. Okay, and this has the enzyme, which is the HRP. It was something like this, I think. Okay, so it will be like this. Okay, so this is how we have to show. So this is actually your detection antibody. Okay, and this is your HRP. Now let's move on to part 2. With reference to 6.3, describe one difference between the capture antibody and detection antibody. So you can just write it as I have also provided you with some of the information. So capture antibody are immobilized while detection antibody has enzyme. Okay, so this is just your answer for this question. Now figure 6.5 outlines the reaction catalyzed by HRP in step 6, the organic substrate TMB. Okay, so now they have told you the organic substance, it is colorless and it is in excess and changes the color when it is oxidized. Now this indicates a positive result. So whenever the TMB changes the color, so it changes from the colorless to blue-green, this means there is a positive result for the test. Now the students suggested that a low concentration of a toxin may be diagnosed as a negative result instead of a positive result. Using a colorimeter after step 6 in the sandwich ELISA test would provide a quantitative result, okay, to help avoid this error. Now with reference to figure 6.5, explain why using a colorimeter would provide a quantitative measurement of a detection of a low concentration of a toxin and help to confirm the positive result. Now actually what happens is that the results, actually whenever there is a color change and we are actually observing it with the eye, so that results are actually subjective, but whenever it is being done with the colorimeter, it gives you numerical values that are not subjective, and it can actually for example and it actually gives you value based on the absorption of light. So for example, if there is less color, there is less you can say there is less color in the mixture, then it will give you low you can say low concentration. And if there is more color, or you can say it has there is less light absorption, then it will give you high concentration. Okay, but if there is nothing, it is colorless liquid, then there will be no absorption, and there is it means that it has no concentration of that given chemical. Okay, so that's actually how the colorimeter works, and that's how you can deduce the concentration of something to help confirm a result. So you can just write it as colorimeter gives non-subjective results as it is based on absorption of light from a reaction mixture. Okay, so it actually is based on the absorption of light from reaction mixture. So if there is less light absorption, there is low concentration and will help show a positive result. Okay, and if there is high concentration, or you can say there is, you can also write if there is a more color intensity, then more concentration of TMB is present. Okay, so if there is more color change, there is more TMB present. So this is how it is going to be giving you the result. And you can also write about that the concentration can be worked out using the intensity of color. Okay, so that's how it is going to be telling you about the concentration of the TMB present and how much the result is towards the positive end. Okay, so if there is no concentration, there is going to be no absorption of light, and the colorimeter will give you that there is no concentration of TMB present. Now this paper is finished. This was a small paper, but there were many parts that were being involved with each of the question. Now if you want a PDF of this paper, what you can do is that simply you can just go to my channel and add into the YouTube bio. Then you can go into the website link and you can click on the website. Then after loading onto the website, what you can do, you can just navigate to this past paper section, and in the past paper section you will find all of the papers for your maths, for your physics, for your chemistry, and your biology. Okay, so you have to go to the May June 2025 past papers and you will find all of the papers here. Okay, so this is how you are going to be getting the PDF of this paper. So this was all for today. If this paper was beneficial, do share this video with your friends so that they also benefit from it. And also, if there are any queries related to this paper or any other question, you can drop them in the comments down below. So this was all for today. I'll be meeting you in another video. Till then, take care. Goodbye.